Yes. Today we'll discuss about the neurophysiology, the main important topic in the animal physiology. First, we'll discuss today what exactly we are going to discuss. In neurophysiology, first we'll discuss about what is nervous system and what are the main parts of the nervous system. After that, we'll discuss about the little bit about the structural components of the nervous system. Then the main structural and functioning of the nervous system, that is called as neuron. We'll discuss about the neuron structure elaborately. After discussing about the neuron structure, we will discuss about the types of neurons because there are different types of neurons based upon their function and based upon their function as well as their structure. Then we'll discuss about the what is that synapse and different types of synapses based upon their structure and their connectivities. After discussing about this synapse, we'll also discuss about the synaptic transmission and mechanism of synaptic transmission. Then, if time permits, we'll also discuss about the membrane potentials, especially while we're discussing about the membrane potential, we'll discuss about the chemical, what is that, and ion gradient in and outside the cell membrane, especially neuronal membrane, especially potassium, sodium, and chlorine, and other, what is that, ions, how they are present extracellular membrane of the neuron as well as intracellular membrane of the neuron. Then we'll discuss resting membrane potential, threshold potential, and action potential with respect to the polarization, depolarization, repolarization, hyperpolarization. These are the main concepts which we will discuss today. Uh, I hope you are understanding my language as well as everything. Is it clear now? Shall I go? Yes. First, we will discuss about nerve physiology and what is the main importance of nerve physiology in vertebrate biology or in vertebrate physiology. Mainly to get the or to give the response against the external stimuli because we are not alone in this environment or in this planet. We are a part of what is that in the environment. We will get the different types of stimulus from the different types of external sources. For example, from your friends, from the environment. For example, you are finding the high cold, but high cold, how you will sensitize? You will sensitize the low temperature and high temperature and temperature differences, humidity differences by our sensory neurons. That means to get the what is that stimulus and give the response against the stimulus from the external environment or external source. That means that source may be anything. If you are giving some response against the stimulus, that is mainly based upon the or due to the neurons or what is that neuronal system. Okay, in nerve physiology, you can see this diagram, functions of the nervous system. What is the main functions of the nervous system? Yesterday, we have discussed about the internal controlling system as endocrine system. Today, we are discussing what is that the main controlling system in higher vertebrates or vertebrates, that is nervous system. If you see the functions of the nervous system like this, you can see because from external stimuli or from external source, we are getting the stimulus here. Your skin may have what is that different types of receptors. These sensory receptors, that receptor which it will take the what is that stimulus from the external source, stimulus from the external source. That neurons we can call it as sensory receptors. These sensory receptors take the signal from the external source and send the signal to this what is that sensory neuron. This sensory neuron will send the signal which is coming from the or signal or what is the stimulus? Signal or stimulus. What is that from external source to what is that central nervous system, mainly brain. Then brain will analyze. That means integrate. What type of information we are getting? For example, you saw a snake. You saw a snake. What you will do? You will afraid. Then you will run. I mean to say that if you want to what is that combat with that snake or if you want to kill that snake, you will show a different type of response. Or if you want to run away from that snake, you will show a different type of response. I mean to say that brain will analyze the what type of information which you are getting from the external source. Then it will integrate. After integration, it will send some signal to the effector organ. But how it will send the signal to the effector organ? For example, you can see here, sensory receptor sending the signal to the, what is that, your sensory neuron? Sensory neuron sending the stimulus to the brain or central nervous system. After integration, type of stimulus or type of, what is that, signal, your brain will analyze. Then interneurons will be there for the time of analyzation. After analyzation, that interneuron will send the signal to the motor neurons to effector organ. I mean to say that effector organ will show. That means it will, you will kill the snake by your, what is that, hands or something, or you will run away from that snake by using your hands. That means after getting the signal, your brain will analyze. After analyzing that analyzed information through the motor neurons, it will reach to the muscle or effector organ. Then it will do its function. Okay. This is about the gist of what is that your central nervous system or the main vertebrate nervous system function. Then what is the anatomical divisions of the nervous system? What exactly you will find in the nervous system? There are three types of nervous system. I kept here only two types, but there are three types of nervous system. Okay, central nervous system, peripheral nervous system, atomic nervous system, not anatomic, atomic nervous system. Especially in central nervous system, we will find the brain and spinal cord. In peripheral nervous system, the nerves which is coming from the brain as well as what is that spinal cord, that what is that nerves which will maintain a or which will prepare a or which will form a system that is called as a peripheral nervous system. The nerves which is coming from the brain, that you can call it as cranial nerves, you can see here. And the nerves which you are finding, what is that coming from your spinal cord, that is called as spinal nerves. I mean to say that the main important integration part of the vertebrate nervous system or higher vertebrate nervous system, that is called as central nervous system, then peripheral nervous system. In central nervous system, we will find the brain and spinal cord. Then how your peripheral nervous system will form? The nerves which is coming out from your brain, the spinal cord, the nerves which is coming out from your spinal cord, that nerves we can call it as cranial nerves and spinal nerves. Both cranial nerves and spinal nerves will form the, what is that peripheral nervous system. Then coming to the atomic nervous system. Atomic nervous system means which is not under the control of your central nervous system, which is under the control of your spinal cord. Okay, that is called as atomic nervous system. That means without any integration, if you want to show, for example, I have thrown a chuck piece on you, then without thinking, within a fraction of a second, your eyelid will close the eye. Okay, for example, you are walking on the road, then suddenly a car came, then you will stop suddenly because there, at that time there is no, what is that time to integrate, taking your spinal cord, what is that sensory neuron, then going to the brain, integration of the brain, then after integration reach to your motor neuron to your effector muscle. This much of time will not be there. Then what will be happen? Your atomic nervous system will take care about the reflex mechanisms. Reflex mechanisms. This is about the atomic nervous system. These are the three main important parts which you are finding in the, what is that structural, what is that division of your nervous system. Okay. Here we are not going to discuss about the brain structure, spinal cord structure, cranial nerves and spinal nerves. Okay. In vertebrate biology, we will discuss that concept. Yes. Then coming to the, what is the main structural and functional unit? Don't forget that this word is very important because every organ system, for example, take the kidney. For example, take the, what is that, brain. For example, take the other systems. Kidney, what is the structural and functional unit of the kidney? We can call it as, what is that, nephron. Like that, what is the structural and functional unit of the muscle physiology? What is that? Sarcomere. Like that, this nervous system is also having the structural and functional, what is that unit that is called as neuron. Okay, that means the neuron is the main base for the structure of the nervous system as well as function of the nervous system. That's why we are calling neuron as structural and functional of the nervous system. Please don't forget this word. Then we'll discuss about the main part of the nervous system, that is a neuron. You can see this diagram. There are different, what is that clumsy diagram you are finding. Actual, the neuron, if you see, if you observe under the electron microscope, that is the same. Okay, you can find the neuron like this. This is actual live diagram of the neuron. You are finding a body and which is having the dendrite as well as axon. That means they are having the branches. I mean to say that the neuron will collect the information or signal or action potential or stimulus from the adjacent neuron. Then after getting the information, it will pass or transmit the new but the same information to the adjacent neuron or other neuron. That's why it is having the branches. It is having the branches. There are two types of branches you will find to transmit or to get the information from the adjacent neuron. Okay, the branches which is collecting are the branches which will take the information from the other neuron. That may be action potential, that may be information or anything. Generally, information in the form of stimulus or in the form of action potential. That branches you can call it as dendrites. You can see here. I hope you are able to see this diagram. Okay, these are the branches which you can find here. This is a cell body. To this cell body, you are finding the branches like structure, finger-like structures. The branches which will take the stimulus from the adjacent neuron and send to the cell body. These branches you can call it as what is that dendrites. You can also call these dendrites as what is that afferent branches. A, not E. Afferent branches means which will get the signal. You can see the red color here. What is that? This signal passage at passing way. Then after getting this information from the adjacent neuron through your dendrites or through the dendrites, it will send the signal to the soma or cell body. That means neuron also a typical eukaryotic cell because this eukaryotic cell is present in the nervous system. That's why we are calling as neuron. If a eukaryotic cell present in the muscular system, we can call it as sarcocyte or myocyte. Like that, this is also a typical, what is that eukaryotic cell? The same properties what you are finding the eukaryotic cell. In cell biology, you will read cell is having some mitochondria, cell is having nucleus, cell is having Golgi bodies, cell is having, what is that endoplasmatic reticulum. The same type of properties you will find for the neuron also. But other eukaryotic cells are having the cell division, but neuron doesn't have the cell division. Can anybody tell the reason why the neuron doesn't have the cell division? Because, sir, you are telling that neuron is a eukaryotic cell, but why neuron doesn't have the, what is that cell division? Any one of you? There are 11 students are there. Any one of you, please tell me that why neurons doesn't have the cell division? Generally, you see that for cell division, we need a main part that is called as a centriole. Okay. Neuron cell body doesn't have the centriole. Please don't forget that. Write it somewhere. This is very important for your competitive exams. The neuronal cell body or cell, okay, you won't find the centriole because you will find the nucleus, you will find the endoplasmatic reticulum, you will find the Golgi bodies, you will find the peroxisomes, you will find the lysosomes, but you won't find the, what is that, centriole. That's why, what is that, the neuron doesn't go to the or undergoes to the cell division. Okay, then you will find the mainly granules. These granules, what is that, mainly having the proteins because neuron mainly useful for the, what is that, the main structural unit for this nervous system because it is having that, because of that reason, it is having the main proteins here. Then after this cell body, you can see here, after this cell body, you are finding a big axon-like structure. Big axon-like structure. Axon-like structure. This is called as a single branch which is coming out or single and long branch which is coming down from your, what is that, soma or cell body. This is called as axon. Okay. The junction between the soma, this one, you can see the diagram, please. The junction between the soma, you can observe my cursor also. The junction between the soma as well as this axon, that is called as axonal hillock. That is called as axonal hillock. And what is the use of this axonal hillock? Sir, why you are telling about the main thing about the junction? Because this junction will maintain the functional property. That means the axonal, what is that, hillock can maintain the, what is that, functional properties of the, what is that, neuron. That's why this is called as axonal hillock. Then coming to the axon, axon is a big branch which is covered by myelin sheet, which is covered by myelin sheet. Sir, why there is a need of myelin sheet? For example, if you see in our homes, we are finding the electric wires. We are finding the electric wires. generally the current will pass. The current will pass in the, what is that, bronze, or what is that, ragi tiga anta manam Telugulo. Bronze, what is
That's
that
true.
wire? On that wire, metal wire, there is a plastic wire. Sir, why you are wrapping that metal wire with plastic wire? Because of only one thing, insulation. Because in one wire, positive will go, in other wire, negative will go. If you combine both positive and negative, there will be a short circuit. That's why to outcome, what is that? To protect from the short circuit, what we'll do, we'll wrap that metal wire with the black and green and red wires. Okay, like that, this neuron also just like a electrical wire, which will pass the action potential, which is in the form of voltage. We will discuss everything today, which is in the form of voltage. That electrical wire, which we are considering as, what is it, comparing with your axon, it should be wrapped. Otherwise, the adjacent neuron action potential or voltage may be disturbed, and there will be a chance of short circuit. That's why what we will, what it will be protected. It is protected or wrapped by myelin sheet. Mainly, myelin sheet is made up of. Mainly, myelin sheet is made up of lipids or fats. Mainly, myelin sheet is made up of lipids and fats. Then one more important thing, who will synthesize this myelin sheet? How this myelin sheet will synthesize are formed on the axon. Mainly there is a cell which you can find on the in the axon, that is called as Schwann cell. That is called as Schwann cell. This Schwann cell synthesize the, what is that, myelin sheet. This myelin sheet is wrapping the axon to works as a insulator. Please don't forget that myelin sheet is made up of fats or lipids. Then this myelin sheet is synthesized by a cell that is called as a Schwann cell. The main use of myelin sheet, insulation. Main use of myelin sheet, insulation, to protect from the, what is that, short circuit with the adjacent axon or adjacent signal. Then coming to the last region, the last ending region, you can see soma is having many branches, but axon is having only one branch. But after in the ending, it is having the different types of, what is it, branches. This is called as synaptic terminal. Synaptic terminal. Then you can also call it as axonal dendrites. Axonal dendrites. They will send the information to the adjacent neuron. That means dendrite will collect the signal. After collecting, axonal hillock will maintain that signal to pass the signal to the along through your myelin. What is that, axon? Then after reaching to the ending of axon, this axon will send the dendrites of the other neuron. Then one more important function which we can discuss here. You can see here myelin sheet is not continuous. Myelin sheet is not continuous. You are finding that, what is that, node-like structure here. One node, two, three, four, five, like this. Why there is a node-like structure? In course of, what is that, evolution, vertebrate evolution, because the electrical potential or voltage which is passing through your axon, it should go very fast, 0.3 millisecond. 0.03 millisecond. You cannot imagine. I'm throwing a, what is that, chakpiece on you. Within a fraction of second, your brain will analyze and everything, and you will close your eye. But how it is possible? Because the signal will pass very, very fast. That is 0.03 milliseconds. That's why to pass this information very fast, the conduction properties should be very much high. That's why, what is that, you are finding this gap. This gaps you can call it as, or this nodes you can call it as Ranvier nodes. These nodes you can call it as Ranvier nodes. This Ranvier nodes, what will be there? The action potential which will not pass very slowly. They will jump from one node to one node, one node to one node, like frog motion. That type of passage is called as saltatory condition. Saltatory condition means jumping condition. Jumping type of movement you will find in the neuron. This is about the, what is that, a typical neuron structure. Then the junction between the two neurons, this is one neuron and this is other neuron. This is called as a synapse. This is called as synapse. Yes, you can see here. This is called as synapse. I mean to say that one neuron is getting the information from other neuron and sending the same information to the other neuron. This junction between the two neurons, axon of one, daily dendrites of one neuron, dendrites of the other neuron, that is called as synapse. Synapse mainly useful to transmit the signal from one neuron to one neuron. This is the basic structure of neuron. Then you can see the same diagram I kept here. In electron, what is that? electron microscope, you can see the, what is that, axon like this. You can see the axon like this. Okay. This is dendrites. This is a nucleus. And this is soma or cell body of this one. Then this is axon. Axon is having the button-like structure. This is called as a axonal terminal, which will pass the information to the next neuron. Is it clear? Is it clear or any doubts?
Sir.
Yes, sir.
Yes, very good. Next. Then just now I said that myelin sheath and Schwann cell. If you zoom this one cell or one node, you can find like this. You see that myelin sheath is not continuous. Myelin sheath is not continuous. This is formed like a node-like structure, and there is a gap, there is one more node, and there is a gap, there is one more node. That means this gap is useful to transmit or jump your information which is passing through your axon, then which will be very fast. Which will be very fast. Here you can see this violet color is called as Schwann cell. This is wrapping in layers. This is called as myelin sheath. Layers of myelin sheath produced by, what is that, Schwann cell, and this is axon. These nodes you can call it as nodes of Ranvier or Ranvier nodes. Then what is that? Neuron interaction and integration. How the neuron will integrate with the effector organ. That is very important. You see here, sensory receptor will send, what is that, collect the information through the skin. After collecting the information, this will pass to the adjacent neuron. This is called as sensory neuron. Sensory neuron to the interneuron, which we are finding in the central nervous system. Then interneurons are the brain, which is brain. They will integrate the signal, how they are getting. After integration, the integrated information or signal will send to the effector organ. You can see here, this is striated muscle, sent to the effector organ through the motor neuron. That means there are three types of neurons which you can find in the nervous system. That is a sensory neuron, interneuron, motor neuron. Sensory neuron will collect the information from external source or skin. Then interneurons will integrate the information, what type of information, positive signal or negative signal. For example, if I give the chocolate, you will smile. This is positive signal. For example, if I scold you, you will cry. This is negative signal. But how you will cry and how you will be like smile, that means that information which will be integrated in your central nervous system. Okay. Then the integrated information has to reach. For example, smiling means your what is it, facial muscles will contract. Crying means your eye muscles will be contract. That means there are some effector organs. Okay. This effector organ will send the, what is it, get the signal from or through the motor neuron. This is about the neuronal interaction as well as integration with the external stimuli and effector organ. Then I don't want to explain this concept, but you plus see, just now I have given one example to you, especially in atomic nervous system. If I throw one chalk piece or suddenly one car came while you are walking on the road, then you will show a very fast mechanism. In that type of condition, there is no sensory neuron, there is no motor neuron, there is no interneuron to integrate in the central nervous system. Just you need very fast response. Otherwise, that car will hit you and you will die. Or your body will be maybe damaged because of that accident. That means you need very fast type of response. That type of response is called as a reflex mechanism. There is a reflex arc which you can find in the body. You can see here, this is called as flexor muscle, which will then this is the spinal cord. The spinal cord is integrating the information and passing very fast without all these concepts what we have discussed. Then what are the different types of neurons? Based upon the structure, neurons are different types. Based upon the structure, neurons are different types. Neurons are different types. There are three types of neurons which you can find in our body. That is unipolar neuron, bipolar neuron, multipolar neuron. Sir, what is this polar? Polar means this is a soma which you can find here. This is a soma. Soma means cell body. The number of branches which is coming out from your cell body, based upon that, neurons has classified into three types. What is that? Unipolar neuron, bipolar neuron, and multipolar neuron. In unipolar neuron, uni means single. Uni means single. Out of this slide, I don't know about the previous slide. Out of this slide, you will get one question compulsory. If you write competitive exams like JL and DL, out of this slides, you may get one question. Okay. If you are getting only single branch, that is called as unipolar. The best example for unipolar, that is the dorsal root ganglion, which you are finding in the central nervous system. Then bipolar, you see, this is a soma, which you are getting only one branch. That branch may be having the different types of branches. You are getting only one extension from the soma. That's why this is called as unipolar. Then coming to the bipolar, bi means two. That means you can see the soma here. This is soma is having the one branch anterior side and one branch posterior side. That's why this is called as bipolar. This branch may have having the different types of branches after extension. That is not important for us. You can see here the soma is dividing or giving the birth for the two branches. That's why this is called as bipolar neuron. Mainly bipolar neuron, you will find the sensory organs. This question came many times in the what is that competitive exams. So the best example for bipolar neurons, that is retina. Here I have written as eye, but retina is the best example for what is that bipolar neurons. And ear, that is the sensory organ. Olfactory info, that means they will collect the information and send the signal to the collect the information from the effector organ and send the signal to the central nervous system. That's why mainly in the sensory organs we are finding the bipolar neurons. Then coming to the multipolar neurons. This is a typical neuron. The neuron which we have discussed, that is multipolar neuron. You can see here. You can see here. This is the soma. Soma is having many branches. Okay, which we are calling as dendrites, which will collect the information. And one more main branch that will transmit the information from soma to other neuron. That's why this is called as multipolar neuron. Most abundant type of neuron because this is a typical neuron which you are finding in the central nervous system. This is about the types of neurons based upon their structure. Yes. There is a one more concept which we can discuss. Okay. Because in many exams where they are asking the questions on the other than the neuron. Because our sir said only neuron, but they are asking about the astrocytes, they are asking about the neuroglia, they are asking about the microglia, they are asking about the macroglia, they are asking about the epidermal cells. What are these cells? That means in nervous system, other than the neuronal cells, there are other non-neuronal cells. There are other non-neuronal cells. That non-neuronal cells, you can call it as glial cells. You can call it as glial cells. Glial cells are non-neuronal cells, mainly useful for to maintain the structure as well as functions of the nervous system, as well as functions of the nervous system. Yes, now we'll see that you can see here the first diagram. Astrocyte, microglia, then epidermal cells, then you can see that oligodendrites, then sensory neuron with the Schwann cell. You can call it as a satellite cell. That means glial cells are different types. Glial cells are different types based upon their function and structure. I will show one more diagram, then it is very easy to understand. You can see. This is about the types of neuroglia. Types of neuroglia. The main glial cell, which they may ask you in the exam. Okay, that is astrocytes, oligodendrocytes, microglia. Sir, what is the use of this microglia? And what is the base for this one? Mainly, microglia means macrophages are dendritic cells, which you are finding in the brain or nervous system. That is called as microglia, which is mainly called as, please don't forget this one. I will give you a gist of what is that macrophages. Microphages, which you are finding in the kidney, that is called as macroglia. Okay. Then the macrophages which you are finding in the brain or nervous system, that is called as glial cells. The macrophages which you are finding in the liver, that is called as Kupffer cells. Like that, every organ is having their own protection to show the immune response against the pathogen. If any pathogen or foreign particle entering into the organ, first you need protection. Okay, that protection non-specifically is given by macrophages and dendritic cells. For example, dendritic cell which you are finding in the skin, that is called as Langerhans cells. Like that, we are finding every organ, we are finding the macrophages to give the immune response or to give the signal to the immune system. We found a affected organ. You please act against that affected organ. Like that, astrocytes are the star shaped. You can see here this violet color, star shaped cells, which will useful to maintain the functions of the, what is that, neurotransmission properties of the neuron. Neurotransmission properties of the neuron. That's why this is called as astrocyte. Then I'm coming to the oligodendrocytes. Oligodendrocytes mainly useful for the structural support. Astrocytes is mainly useful for the, what is that? Functional support. Oligodendrocytes mainly useful for the structural supports. For example, Schwann cell. Schwann cell is maintaining the myelin sheet in the peripheral nervous system. In central nervous system, these oligodendrocytes are useful to synthesize or form the, it is that myelin sheet around the axons. The microglia is just like protective cells which will collect the foreign particles as enters into this brain. That's why this is called as microglial cells. Then epidermal cells and satellite cells. Already you know about the Schwann cell. This one, this is called as Schwann cell. This yellow is called as myelin sheet. Satellite cell and epidermal cells. Okay, they are having a group of Schwann cells which is useful to maintain this. What is that? Your myelin sheet. That's why this is called as satellite cells. Both satellite cells and peripheral, what is that? Schwann cells you will find in the peripheral nervous system. Oligodendrocytes, active astrocytes, microglia, and epidermal cells. Ependymal cells which you will find in the, what is that, central nervous system. This is about the types of glial cells or non-neuronal cells which you can find in the central nervous system as well as peripheral nervous system. Then we will discuss the main concept. Now we have discussed about the what is nervous system, what is the components of nervous system like central nervous system, and what is that peripheral nervous system. Under central nervous system, we have discussed about the brain as well as spinal cord. In the peripheral nervous system, we have discussed about the nerves which is coming from the brain, that is called as cranial nerves. Nerves which is coming from the spinal cord, that is called as spinal nerves. Then we have discussed about the structure of neuron. We have discussed about the structure of axon, then types of neuron, and types of glial cells or non-neuronal cells and their function. And this is a basic part of the nervous system. This is the basic part of the nervous system. But if you go to the depth in the nervous system, where we can discuss about the different types of potentials. Sir, first, what is potential? What is potential? Before going to discuss about the potential, what exactly we'll discuss in the measuring the membrane potential? What exactly we'll discuss in the measuring the membrane potential? There are three important points which we can find in the membrane potentials of an axon that is called as resting membrane potential. The potential which you are finding in the potential which you are finding in the when a cell is the resting stage or when a neuron is a resting stage, that is called as a resting membrane potential. We'll discuss later. Then next we'll discuss about the threshold potential. Next we'll discuss about the threshold potential minus 55 millivolts. Then we'll discuss about spike potential or action potential. Spike potential or action potential. Before discussing about these membrane potentials, we will discuss about what is potential. What is potential? Can anybody know what is potential? Krishna, Mugileshwari, Sairam, Redmi, Shashikal. What is potential? Potential means ionic current. Ionic current around the cell membrane, that is called as potential. For example, if you go a electrical substation, the potential will be more. If you touch the electric wire, then it will be like ash. For example, if you touch a small wire which is going in the in your home, if you touch, then you may get the shock, but you won't die. Sir, why? That is also electric wire and this is also electric wire. Because Any doubt? Arnavi, Aniravan Mamidi. Any doubts?
No, no, no.
Yeah. If you have any doubt, you can stop me. No problem. Because this is not, it is that man-to-man class. Okay. That's why you may have many doubts. After class also, you can discuss with me. No problem. That means the electrical potential will be different with reference to the different types of areas. Like that, same thing in our body also. In a neuron, ionic current will be there. Sir, what is the meaning of current? Current means movement. Current means movements. That means movement of ions for a particular stimulus at a certain area. That means when
ఇక్కడ
you are
చెప్పాను
given,
కదండి క్లాస్ అయిపోయిన తర్వాత
what happened?
అంటే మీ ఇష్టం
What happened? Who is that?
ఆయన పోస్ట్ డాక్ బాగా చెప్తున్నాడు.
What is this ionic current? Ionic current means ions will move from inside to outside sometime, inside to outside sometime, and outside to inside sometime. This is called as ionic current. Then that means the ionic current property of a membrane. For example, mainly uh, all membranes doesn't have this type of current properties. Especially there is a mechanism, there is a concept that is called as electrophysiology. There is a concept that is called as electrophysiology. That means there are three types of excitable cells. There is a there are three types of excitable cells in our body. The first type of cell that is called as what is that neuronal cell. Second type of that is called as muscle cell. When we are in MSc, I think 15 years back, we used to read excitable cells only two types. Now they have added one more cell that is endocrine cell. I mean to say that excitable cells are in our body. There are three types that is called as neuronal cell, muscle cell, as well as your endocrine cells. That means, sir, what is excitability? What is excitability? Excitability means when you are giving some stimulus, it will show some response. This is called excitability. But why you are finding this excitability in these cells only? Because these cells are having, especially neuronal cell, muscle cell, as well as your endocrine cell, they are having the current properties. That means movement of ions in and out due to the signal or stimulus, due to the signal or for the signal and what is that and stimulus. Okay, first we will discuss about the what is this ions, how these ions are present in and outside the cell membrane. For example, you can see here this diagram. Here you are finding the yellow color line. This is called as plasma membrane. This is called as plasma membrane. I hope you are seeing this diagram. This is axon. I am taking a small part of this axon. This axon is having the, what is that, like this. Axon is having the plasma membrane. Then inside the plasma membrane, you can call it as intracellular fluid. Then outside the plasma membrane, you can call it as extracellular area. Then what are the different types of ions you will find inside the cell membrane or outside the cell membrane? Before discussing about the ion concentration, we'll discuss about the charge. We'll discuss about the charge and we need to discuss about the how this charge will come. You can see here, this is cell membrane. Don't see this voltmeter. Don't see this voltmeter now. We'll discuss about this voltmeter properties after some time. First, you can see here, outside the cell membrane, you are finding the positive charge. Positive charge means cationic charge or anionic charge.
Cationic charge with the help of sodium ions.
Okay, okay, great. Why sodium is more in the outside?
It is the extracellular electrolyte. It is which is present in extracellular.
Why?
In the endoplasmic reticulum, the sodium ions may be present. I think so.
Outside, you won't find the endoplasmic reticulum. Inside, you will find the endoplasmic reticulum. And endoplasmic reticulum is mainly having the calcium. It doesn't have sodium. Very simple. Okay. I don't know your name. What is your name?
Sir, this is Raju.
Raju. Raju, the answer is right. The outside, what is that? You will find the sodium ions. Because why you will find the sodium ion? That is very important. In course of cellular evolution, like we will discuss as invertebrate evolution, vertebrate evolution. But because we are discussing about cell here, we are discussing about only cell ions around the cell. In course of cellular evolution, the cell is evolved mainly in the sea. That's why cell mainly having the, or what is that? If you take the cell here, around the cell, what you will find at the time of evolution, you will find the sodium ions. You will find the sodium ions. That is the reason you will find what is that more sodium ions outside the cell. Then what is that? Due to this more sodium ions is a cationic charge. That's why you will find the positive charge. As Raju, you can give the answer for this question also. Not only Raju, anybody can give the answer. Uh, if I'm sorry because if I'm asking only Raju because I don't know your names here. Okay, then you can see inside. Inside you are finding the negative charge. Why?
Chloride ions.
Okay, what is very less chloride ions? You won't find chloride ions more. You will find very less chloride ions. More or less chloride ion concentration will be same in and out. Next. Any one of you? Generally, while we are discussing about these properties, yes, in the diagram, what I have drawn that what is that positive charge outside, as Raju said that sodium ions are more in the outside, then potassium ions are more in the inside. Potassium ions are more in the inside. But the problem, potassium also cationic charge. It is not a negative charge.
SS K plus ion.
K plus ion. But how come you will find the negative charge? This is very important. This is very important because this is inside the cell membrane. Outside the cell membrane, you will find only water or you will find extracellular fluid where you will find chlorine, where you will find potassium, but very less concentration. You will find sodium, more concentration. But coming to the inside also you will find sodium. Inside also you will find the chlorine. Inside also you will find the potassium. But potassium concentration will be more or less 150 millimolar. But how you will get the negative charge? Because of only one reason. Mainly, you will get the negative charge due to the organic molecules. Due to the organic molecules like proteins, like phosphates. You see that ATP, take the ATP. You won't find the ATP outside your cell, but you will find the ATP inside your cell. But what is the charge of ATP? Charge of ATP, what is that?
Negative
Phosphate
charge.
groups. Highly
3
positive.
phosphate.
What is that negative? Not negative, highly negative. What is the charge of your amino acids? Many amino acids are having the negative charge.
Negative charge.
Okay. As
Aspartic
you
acid,
said that,
glutamic acid.
you can also add your chlorine here. You can also add your chlorine. But chlorine is not only the reason to have the negative charge inside the, what is that negative charge inside the membrane? That means organic molecules like proteins, fats, then what is that? Your proteins, then all these molecules is the main cause for the negative charge. Yes, you can see the next diagram. Here you can find the what are the main, what is that ratios of ions you will find. Inside the cell, potassium 150 millimolar. Please don't forget, this is very important. Okay, inside the cell, potassium ion concentration, I have written here 150 millimolar, but if you see what is that Fox book, you will find 146 millimolar, but more or less 145 to 150 millimolar. Okay, sodium ions 15 millimolar, chloride ions 10 millimolar. What is that? Other organic molecules. See, A minus. A minus means negative charge, more or less 100 millimolar. More or less 100 millimolar. Then ATP molecules, phosphate groups, your DNA and RNA. What is the charge of DNA and RNA?
Negative charge.
Negative charge. All
Due
these
to
molecules
phosphoric
are,
acid.
yes, because the backbone of your, what is that? This molecule is having that negative phosphate groups.
Diester linkage.
Yes. That because of that reason it is having the negative charge. I mean to say that inside a cell you will find, yes, when we're discussing about potassium ions, you need to have positive charge. But inside cell, other than the potassium ions, you are finding the organic, inorganic, phosphate groups, ATP, many things are there. That's why you will find the negative charge. Then coming to the outside, this is outside the cell membrane. You can see outside. You can see just now Raju said that sir, more chloride ions, but chloride ions are more in the outside. When sodium is there, 100% you should find that what is that chlorine. Then sodium ions more. Potassium ions are very less. 5 to 6 millimolar. Chlorine ions 120 millimolars. Sodium ion concentration will be more. I mean to say that why I am showing this diagram. What is the importance of this diagram? That means when you are given some stimulus, there are some channels which will open. There are some receptors or channels will open. When you open one channel, for example, here I have given a stimulus. If you open this, what is that sodium channel? If you open the, so for example, that stimulus is opening the sodium channel. Sodium will be influx or outflux. Any one of you think that I have given a stimulus. When I have given the stimulus, sodium will influx into the cell or outflux out the cell, because you cannot say that sodium, sir, there is no sodium. Here you are finding the sodium. There may be a chance of outflux or there may be a chance of influx. But my question is very simple. When you are given the stimulus on the neuron, sodium will be influx or outflux?
Influx, sir, because
Influx,
the
influx.
concentration of sodium is higher
Excellent,
in
excellent. Very good, very good. Because movement of ions based upon the chemical gradient. Based upon the chemical gradient. If you see the Fox book, he has given like this: movement of ions around the cell membrane based upon the electrochemical gradient are under. He will talk like this. I mean to say that the movement of ions based upon the electrical property, because I have given the stimulus, then the channel is opening. After opening the channel, due to having the more sodium ions inside the cell membrane, sorry, outside the cell, then sodium ions will enter into the inside the cell membrane. That means sodium ions influx into the cell. Sodium ions influx into the cell. The same thing. If I have given a stimulus, then what about the, if you open the potassium channel, what will be the scenario? Then potassium ion
Inside to
concentration
outside.
will be, yes, excellent. Potassium ion concentration will be more in the inside. That's why less in the outside. That's why they will be outflux. Please don't forget this basic concept. Total
Sir, is
generation
it
of,
sodium potassium pump?
no. Sodium potassium pump is different.
Okay.
Sodium potassium pump is different. Okay. We will discuss that sodium potassium pump also after some time.
Okay.
But there is an individual sodium channel. There is an individual potassium channel. You can see this diagram. Next diagram. This is potassium channel. This is sodium channel. You can see, you are seeing this diagram now. This channel is called as sodium potassium pump. And don't forget that the first two channels will follow the electrochemical gradient. But sodium potassium channel will not follow the electrochemical gradient. This is active transport which will take the energy, because generally what I can say that potassium will be outflux, sodium will be influx. But you can see this diagram. That's why I don't want to confuse you. Don't discuss now the sodium potassium channel, because here sodium will be outflux and the potassium will be influx and it will use the ATP. But in these first two scenarios, it won't use the energy. This is just electrochemical gradient. That means being being a MSc student, you please keep it in your mind. When you open the sodium channel, sodium will be influx. When you open the potassium channel, potassium will be outflux. Then coming to the sodium potassium channel, this is totally different, opposite to this main mechanism. You can see here, sodium potassium pump. 2 potassium ions are coming in, 3 potassium ions are coming out. This is a different scenario, and by using the ATP energy. But here, this is simple diffusion. It won't use the energy. It won't use the energy. Okay. Then, sir, why you are telling like this? Because to explain about the membrane potentials. To explain about the membrane potentials. That is resting membrane potential, threshold potential, action potential. You need to know the ion movement or ionic current. Ionic current. Now we'll discuss what is potential. Then we'll discuss resting membrane potential. Then we will discuss threshold potential. Then we will discuss action potential. Then we will discuss depolarization. Then spike potential. Then repolarization. Then hyperpolarization. Okay. These are these main concepts which we can discuss in the nervous system. I hope next diagram is there. Yeah. This is very important graph. I think triangle graph. If you write any exam like JL and DL, you will get one question from this diagram. 100% you will get one question. Many times they have asked a question, what is threshold potential? And what is the amplitude of threshold potential? Okay. Last time in set and net, they have asked one question. You are given the, please listen carefully. The question is like this. You are given the stimulus. it is depolarized with the cell membrane. At the time of depolarization, this is 2 marks question. At the time of depolarization, what type of channels will open on the cell membrane? A. Answer is like this. A. Voltage-gated cationic channels. B. Voltage-gated anionic channels. C. Ligand-gated cationic channels. D. Ligand-gated anionic channels. What is the answer? Any one of you.
Sir, voltage gated cationic.
Why voltage-gated channels? Why not voltage-gated anionic channels?
So that depends on voltage. Maybe.
Yes, one student sent one message. First, we'll discuss that one, sir. What is potential? What is potential? Potential means ionic equilibrium. Ionic equilibrium or ionic strength of the ionic current around the cell membrane is called as potential. Strength of the ionic current. Current means movement. Strength of the ionic current around the cell membrane, in and out. Around means in and out. That is called as potential. That is called as potential. Then this time we have discussed about one question. What is that? When we are given the stimulus, okay? Yes, as she said that the answer is right. That is called as voltage-gated cationic channel. We'll discuss why this is voltage-gated cationic channels. Okay. Now, I hope you are able to see this diagram, refractory period. You are seeing this diagram?
Yes, sir.
Yes, sir.
Yeah, thank you. Now I am maximizing this maximum. We will spend here for 15 minutes. Okay. First, we'll discuss. Now we know that what is potential. What is membrane potential? Then we discuss what are the different types of membrane potential which you will find in the neuronal cell or excitable cell. There are 3 types of membrane potentials. Resting membrane potential, the potential difference. Listen carefully. The potential difference around the cell membrane or potential equilibrium around the cell membrane when a cell is in the resting stage is called as resting membrane potential. That means there is no any stimulus to the cell, then the cell will be in the resting stage. The cell will be in the resting stage. This is called as resting membrane potential. Here you can see this blue line. There is no any action potential. There is no any action potential from this point to, that is 4 to 7. In this area, there is no any potential. There is no change in the graph. That's why this is called as resting membrane potential. Then what is the amplitude of the resting membrane potential? That is very important. That means generally many students, give the answer like this. What is the amplitude or strength of the resting membrane potential? Sir, it is in the resting stage now, sir. That's why this is 0 millivolts. Generally potential we will measure in the form of millivolts or volts. Okay. Generally it should be like what is that 0 millivolts. Yes or no? But why? You can see in the diagram, this is more or less minus 70 millivolts. If you take the skeletal muscle, that is minus 65 millivolts. If you take the cardiac muscle, that is more or less what is that minus 70 to minus 80 millivolts. Sir, why it is in the negative? Can anybody give the reason? Because it is in the resting stage, it should have 0. But why it is having minus 70 millivolt? No problem. Raju, any answer? No problem. If it is wrong also, no problem.
Due to the less potassium ions.
Less potassium ions inside is more, no? More or less 150.
Actually, it is if it is a resting stage,
Yeah.
outside more potassium, sodium ions. Inside, compared to sodium, there be a less potassium ions.
No. Okay. You came near, but the answer didn't came out that what is formed exactly. Just you see that. Generally, sodium channels are very tight channels. Sodium channels are very tight channels. It is not that much of easy to leak, but potassium channels are leaky channels. Potassium channels are leaky channels. That means, for example, if I ask you, you please, there is a tap which is coming down, or which is water is coming. Then I have asked you, Raju, you please close the, or what is that, switch off the tap. Then you will switch off the tap, but you are not switched off perfectly. Then what will happen? The drops of the water will be leak. Same thing. The potassium channel, which you are finding on the cell membrane, it is leaky in nature. But the sodium channel, very fast and very tight. That means when you are given the stimulus, they will open very fast. Sodium influx will be very high, but potassium channels are very slow, and at the same time, they are leaky in nature. When there is no resting stimulus, the cell is in the resting stage, then sodium channels will be very tight. There is no chance of leakage of the sodium. But potassium channels are leaky in nature. In resting stage also, less number of potassium channels, what is that, leak from the cell. That is the reason, or that is one of the main important reason, what is that, which is having the negativity or negative charge when a cell is in the resting stage. That's why if anybody asks you, what is the resting membrane potential? Resting membrane potential is minus 65 millivolts to minus 70 millivolts. Minus 65 is an excellent answer. Okay. Minus 65 millivolts to minus 70 millivolts. Okay. Because you can see here, I'm not changing the diagram. I'm keeping the diagram. What is that? A simple line. But if there is any, like, what is that, graph like this, then you can say that there is a change. Just like, what is that, a small line, which is not, there is no any disturbance. Then you can say that is a resting stage. Then what is threshold potential? What is threshold potential? Yes, now you are given the, here you can see that red mark. I'm giving the stimulus here. I'm giving it this, but this is resting membrane potential. At first minute 1, what I have done, I am giving the stimulus here. While I'm giving the stimulus here, then you found the, what is that, a gap, what is that, a rise in the graph. But you are not calling directly as action potential, you are calling as threshold potential. For example, I will give a small example to you. Annual physiology students are not coming to my class. Then what I have done, I have started the, if you come to the class, I will give 1 chocolate. Think like that. Please don't laugh because you are MSc students. But to explain the, what is the threshold potential, I am giving a simple example. To annual physiology students are not coming to the Sridhar sir class. Then what I said that, if you come to the class, I will give 3 chocolates to you. Then Raju said that, sir, we are MSc. If you give the 3 chocolates, we won't come. Then what I have done, I have increased the number of, what is that, chocolates. Then what I suddenly what I said that, if next time if you come to the class, I will give 10 chocolates. Then many students came. I mean to say that to get the response to attend the class, to my class, what I have done, the minimum number of chocolates, that is 10. That is 10. I mean to say that threshold potential means, please listen carefully. Threshold potential means the minimum potential or stimulus strength to get the action potential, that is called threshold potential. That means if it is below minus 55, it won't generate the action potential. If it is above minus 55, it will generate the action potential. That means if you are given a small stimulus, it won't generate. If you are given a stimulus which can generate the, what is that, action potential, that is called as threshold potential. I mean to say that to get the action potential, you need a minimum strength or minimum potential, that is called as threshold potential. You can see this dotted line, that is minus 55 millivolts. At minus 55 millivolts, so you are finding the action potential. You are finding the action potential. Now we see that what exactly happening when you are given the stimulus in the neuron or axon. You see that, yes, you are given the exact stimulus that is above your threshold area. Threshold area means if it is above threshold area, then it will, what is that, it will come out. For example, you see that we will see that opening the gates in the project. What is that? It needs threshold area. After reaching the threshold, if it is more than the threshold area, they will open the gate. For example, if you go to the Srisailam project, what is that? The maximum length of the project is 850, what is that, feet. After reaching the 800 water length, then they will open the gate. Then the water will come out, then the turbine will run, then electricity will be generated. Like that, there is a minimum strength or minimum potential you need to generate the action potential in the neuron or axon. Then we'll discuss three letters first. What is that? Depolarization, repolarization, then hyperpolarization. You see here, in this, after above this threshold potential, you are finding a spike potential. This spike potential you are calling the action potential. You can also call this a spike. It is when it is raising. This is called as a depolarization. When it, after reaching spike potential, that is plus 32, plus 35 millivolts, then it is coming down. That means this is called as a repolarization. Then it is more than the minus 75. It is coming down more than or below the resting membrane potential. This is called as hyperpolarization. This is called as hyperpolarization. After some time, it is reaching to the polarized condition. First, we'll discuss what is polarization, what is depolarization, what is repolarization, and what is hyperpolarization. How a neuron is depolarized, how a neuron is hyperpolarized, how a neuron is repolarized. First, we'll discuss what is polarization. Polarization means ionic equilibria. Ionic equilibria around the cell membrane. You can see this diagram. Plus, minus, plus, minus, plus, minus. That means there is an ionic equilibrium. There is a ionic equilibrium around the cell membrane. This is
The
state of the
number
cell
of
membrane.
cations are equal to the number of anions.
Yes, yes, that is called as ionic equilibrium. Raju, yes or no? Okay.
Yes, yes.
Yes, the number of ionic equilibria means number only. Ionic equilibria around the cell membrane, in and out, that is called as polarization. Polarization. Depolarization. What is depolarization? Depolarization means ionic
Removed.
disequilibrium. Listen carefully. Ionic disequilibrium. Reasons may be different. Reasons may be different. But listen carefully. Polarization means ionic equilibrium. Depolarization. D means you are disturbing the polarized condition due to the different types of reasons. You are disturbing the polarized condition. That is called as depolarization. Depolarization means ionic disequilibrium around the cell membrane. For example, you have opened the channel, then more sodium ions influx, then cationic charge will be increased, anionic charge will be decreased. This is called as ionic disequilibrium. This ionic disequilibrium leads to the cell membrane. That condition is called as depolarization condition. Then repolarization, that means every time in the cell, because cell is in the nature, okay, in the nature, every time the cell will try to, that is a polarized condition, resting stage. Every time cell is, it want to be in the resting stage. It doesn't want to be in the excited state. Manam, we are also, we will get angry for a certain time, but we won't get the angry for 100 minutes and 200 minutes. For a fraction of a second, we'll get the anger, then we'll come down to the base. Okay, no problem, sorry. Like this, we'll come down, like come back to the resting stage. Like that, every cell in our body will try to be come down to the resting stage. To come down, it should be like, what is that? Repolarize. That means it want to equilibrate the ionic gradient around the cell membrane. This condition is called as repolarization. Hyperpolarization means more polarized condition. That means you can see here, this diagram. Hyperpolarization means it is coming to the depolarized condition going up, spiking. Repolarization means coming down. But it should stop here. At minus 70, it should stop. But it is going down below the minus 70. This is called as hyperpolarization. But it is very, very less. That is more or less 0.8 and 0.9 seconds, 0.9 minutes. Then it is milliseconds. Then it is come down, come back to the threshold. What is that? Sorry, resting membrane potential. This is called as repolarization. These three concepts understood, everyone?
Yes, sir.
Yes, very good. Now we will see that how the cell will be depolarized, how the cell will generate the action potential, how the cell will be repolarized. Then what is the reason for hyperpolarization? Why not it is stopping at what is that resting membrane potential? We will discuss now. Yes, I have given the stimulus. Please listen carefully. I have given the stimulus at this point. Then it is more than the threshold potential. What will be happen? When you are given a stimulus, think that this is a neuron. I have given the stimulus here. I have given the stimulus here. What exactly is happening? How it is depolarizing? How it is, what is the disturbing the polarized condition or ionic equilibrium? At that point where you are given the stimulus, For example, I have given the stimulus here. What will be happen? Sodium, I told that sodium channels are very, very fast channels. Sodium channels are very, very fast channels. What will be happen? Sodium channels will be open at that point. More sodium ions. I told that when you open the sodium ions are influx or outflux?
Influx.
Influx.
Yes. Sodium ions will be influx more. When sodium ions are influxing more, what will be happen? Generally, inside the cell, you are finding the negative charges. But
Yes, sir. It
more
may become
sodium
a
ions are
positive.
influxing, then cationic charge will be increased. Cationic charge will be increased. When cationic charge is increasing, what will be happen? Then it will be depolarization. Very fast and high. That means I told that sodium channels are very fast active channels which you are finding, active ionic channels which you are finding on the cell. Then more sodium ions will be influx. Then there is a rise in the action potential. Then there is a rise in the, what is the depolarization. This rise up to the, what is that, plus 35 to, what is that, plus 32 plus 35 millivolts. If anybody asks you what is the action potential or spike potential, that is plus 32, 35 millivolts. Plus 35 is the excellent answer. If you see that your what is that Open University book, you will find there plus 35 or plus 30 is the excellent answer which you can give. Then it will reach the action potential. Yes. Sir, when it will be stopped? That is very important. Yes, sodium influx will be more, but when that sodium channels will be stopped? When the sodium channels will be stopped or closed, the signal strength will be decreased after some time. For example, you are sleeping, then I pinched. At the time of first second, the pinching, what is that, pain will be more. But after some time, the pain will be decreased. Same thing. At this point, I have given the stimulus. Then suddenly you found that sodium influx, more sodium influx. After reaching certain area, then the signal strength will be decreased. When signal strength is decreasing, I told that sodium ions are active in opening as well as closing. They will close very fast. After closing, what will be happen? This sodium influx will be closed. Then I told that potassium channels are very lazy channels. Then to repolarize the cell, what will be happen? Potassium channels will be open. If you open the potassium channel at this point, what will be happen? Influx or outflux?
Outflux.
Super. Potassium ions will be outflux. Potassium ions will be outflux because potassium ion concentration will be more inside the cell than the outside. That's why positive ions will be coming out. Due to the sodium influx, more positive cationic charge are, what is that, jammed in the inside the cell. Then due to the outflux of potassium ions, slowly, cationic charge will be taken out. I told that potassium channels are very, very leaky channels as well as very lazy channels. After reaching resting membrane also, this is called as repolarization. After reaching resting membrane potential, it should stop. But I told that they are very lazy channels. They cannot close perfectly after reaching at this resting membrane potential. After reaching certain down, it will close. Then it will come back to the resting stage, the resting membrane potential. This lag, that is 3 to 4 milliseconds, this is called as hyperpolarization. This is called as undershoot. This is upper shoot. Okay. This you can call it as, this is about the action potential generation or spike potential generation in the form of voltage, like what is that, minus 72, minus 55, minus 55 to minus 30, sorry, plus 30, plus 32, once again, minus 70, where we can, you should know that what is resting membrane potential, what is potential difference, and what is ionic current, what are the different types of ion channels which you are finding on the cell. Then at the same time, what is the depolarization, what is the repolarization, what is hyperpolarization, what type of channel. You can see that she given answer that voltage gated cationic channel, because here we are given the stimulus, we are not given any chemical or ligand to generate the action potential. That's why that is voltage gated. It is opening the sodium channel. That's why this is cationic channel. Here also, when it is coming after reaching the action potential, which is coming down. Here also it is opening the potassium channel. Maybe you can hear influx, here outflux. But this channel also you can call it as a voltage gated cationic channel. Then it is coming down, it will reach to the resting membrane potential. Any doubts here?
Sir, what is the volt of repolarization, sir?
Because there is no exact voltage, Sairam.
Sir, okay.
There is no exact, because just this is what is that, this is very, very less. That is, maximum it will come down up to minus 85, maximum. Okay. Generally coming to what is that? I told that here the diagram is minus 70, but exact resting membrane potential in the skeletal muscles, you will find minus 65 millivolts. It can come down minus 80 to 85 millivolts only. Then the cell never wants to be like what is that, hyperpolarization or hypopolarization. That's why it has to come back to the resting membrane potential. Okay, that is based upon the neuron or axon. Okay, that's why there is no exact any strength or any voltage for this hyperpolarization. Maximum minus 5 to minus 10, comparing with this minus 70. Okay. Any other student doubts?
Sir, in depolarization only sodium channel is
Yes.
open.
Yes, in depolarization, only sodium channels. Why? Because I told you now, they are very active. They are very active. More influx will be there. Then after reaching to the spike potential, when stimulus strength is decreasing, sodium channels are very fast. They will close. Then the lazy channels which you are finding in our cell membrane, that is potassium channels, they will open. When you are opening the potassium channels, cationic charge will be outflux. Then it is coming down. That is called as repolarization. Okay. Any other
Thank you.
students? Please ask any doubts, because this is very important. Because here names are very less. Galaxy 7 Max, something. Mobile names are there. Redmi. That's why please, if you have any doubts, you can ask me or you can message me, no problem. Because we are jumping into anyone, please.
Sir, every time sodium ions are entering into the inside, no, sir. How the sodium ions are get outside after completion of this action potential, everything?
Yes, after completing the action potentials, are more sodium ion will be there in the cell. Okay,
Yes, sir.
you can ask that question, Raju.
Yes, sir.
Osmolarity. Okay.
Okay.
Okay, osmolarity, based upon the osmolarity, slowly after reaching the cell, the sodium ions will be used in your body. Okay, it may go out from your body through your water. It may used in the different types of metabolic reactions, because sodium is very essential. What is that, ion which you can which is useful for the different types of metabolic reactions. It may go out from your what is that, nitrogenous, as a nitrogenous waste metal. For example, if you see our urine, mainly you will find that sodium ions, okay, as well as potassium ions. Yesterday we have discussed one concept, ADH, antidiuretic hormone, because
Yes, yes, sir.
okay, the cellular metabolic reactions, in cellular metabolic reactions, mainly what will be formed? Water as well as carbon dioxide. Carbon dioxide is a different concept, because it is taken out through your transport of gases. But you can see nitrogenous waste material in the water, which is taken by your portal system or your venous system, and taken to your what is that, nephron. After reaching to the nephron, the sodium ions will be, and potassium ions and chloride ions will be re-uptaken to your portal system by ADH. That's why we are calling that is as antidiuretic hormone. Okay, that is very important. Okay,
Vasopressin.
vasopressin. Yes. Next, anyone?
Excuse me, sir.
Yes, tell me, what is your name?
Sir, Mogeshwari, sir, my name is. In
Cheppandi.
this topic, what type of questions it will come to exam, sir?
For your theory exam, mainly
Yes, sir.
you can ask that types of neuron. For short
Okay.
question, what is neuron structure? Then they will ask mainly short question. They will ask what is that, what are the different types of membrane potentials which you will find in the axonal membrane or excitable cell. Okay. Then they
Okay,
will
sir.
ask then what is that,
Synapse.
generation of action potential. Generation of action potential.
Okay.
These are the
They
topics
will
which
ask
you will
like this
get.
also, I think so. Maybe that.
Yes.
Okay, sir.
Then we will discuss about the next main important topic. Actually, time is very only 10 minutes. That's why, but we will discuss at least about the synapse. Okay, then next class we'll discuss about the synaptic transmission. Okay, we'll discuss synaptic transmission in a different way because we read the synaptic transmission in 10th class or intermediate and degree. But at PG level, we'll discuss synaptic transmission in a different way. If you are having a book that is called as Medical Physiology by Ganang, try to read that book about the synaptic transmission because many times the question came this on synaptic transmission like this. What are the different types of proteins, anchor proteins, which you are finding in the synaptic transmission, which we are not finding. Yes, we are finding the same concept in the Open University book, but we are not finding in other books, like Telugu Academy books or intermediate books. Like what is that synaptogen, synaptin, synaptobravin, synaptin. These are the different types of proteins which we are finding in the, what is that, presynaptic bud, which is used to anchor your synaptic vesicle to the presynaptic membrane. We will discuss very elaborate about the synaptic transmission. First, we'll discuss what is the synapse, what are the different types of synapses which you will find in the body. Then we'll discuss the synaptic transmission. After discussing about the synaptic transmission, we will discuss what is that, what is EPSP and what is IPSP. These are the topics. Then finally, we'll discuss the last concept that is called as neurotransmitters. That means at least it needs 2 hours class, that means 3 hours class to complete this neurophysiology. Okay, then we will see that we'll jump into that concept. Yes, nerve impulse, propagation of nerve impulse or conduction of nerve impulse. Then that means conduction of nerve impulse means passing of your what is that action potential from one neuron, one place to one place, one place to one place. That is called as a conduction of nerve impulse. Generally, nerve impulse conduction in the vertebrate neuron or higher vertebrates, that is called as saltatory conduction. Saltatory conduction means jumping. You see that red arrows here. Red arrows says they are jumping from one node to one node, one node to one node. They are not passing like this continuously. So why there is a jumping like that? Because I already told that in higher vertebrates, there is a need of fast nerve conduction. Fast nerve conduction because we are not sedentary animals, we are active animals on this planet. That's why in course of vertebrate evolution or vertebrate physiology, there is a change in the myelin sheet structure in the central nervous system as well as peripheral nervous system. That's why this you see that in myelinated neurons, only unmyelinated regions of the axon depolarize. That means why they are not depolarized? Because this is fat layer where you won't find the movement of ions. That's why unmyelinated area only you will find the depolarization. That's why what will be there? This is like what is that protective mechanism to increase the conduction speed of what is that what is the action potential from one neuron to one neuron. You see here, this is myelin sheet, this is distal axon, and this is proximal axon. Then what is the passage of your action potential from one node to one node? That is the use of Ranvier node. This type of jumping conduction property, you can call it as saltatory conduction. This type of saltatory motion you will find in the frog, in the amphibia. Okay. Then coming to the synapse. Synapse means, what is that? Okay, these are the different types of synapse. First we'll discuss about the synapse. Synapse or synapse means the gap or the junction between the two neurons. The gap or junction between the two neurons is called as synapse. For example, this is axons of the one neuron, dendrites of the other neuron. But there is a gap. That type of gap is called as synapse. That means junction between the neuron and neuron, that is called as synapse. Yes. Sir, why there is a different types of synapses? You said that there is a gap between the two neurons or junction between the two neurons, that is called as synapse. But why there is a what is that different types of neurons? Based upon the structure, there are three types of synapses. Based upon the function, there are two types of synapses. Now we will see what is structural based classification of the synapses. Yes, if the junction between the, what is that? Synapse means junction between the two neurons, that is called as synapse. If the junction between the axon of one neuron, dendrite of the other neuron. Axon of the first neuron, dendrites of the second neuron. That is called as axodendritic synapse. You can see here. You can see here the red color one, neuron 1, blue color one, neuron 2. I hope you are able to see this diagram.
Yes,
Yes,
sir.
sir.
Yes.
Yes, very good. Okay, axon of the one neuron and dendrites of the, this is axon, red color, dendrites of the other neuron, making a junction. That's why this is called as axodendritic synapse. Generally, the synapse in the nervous system will find this type of things. But you cannot say that. For example, if you go to the central nervous system, you will find junctions of the network of the nervous system or network of the neuron. Network of the neuron, where you may find the other types of synapses. That means, what is that? Axon of the other neuron,
Cell body of another neuron.
cell body of the other neuron. That is called as, I already told that cell body, you can call it as soma.
Soma.
That is axosomatic synapse. Axosomatic synapse. Then you can see that axon of the one neuron,
Another
axon of
neuron.
the
Axon.
other neuron. That is called as axonic synapse. This is how classification of synapses based upon the structure. Classification of synapses based upon the structure. In everywhere, transmission or conduction of the nerve impulse is same, but junctions are different. But at MSc level, we can discuss this concept only. Okay. Then what is that? And what I said that this is structural based. What about functional based classification? What about? I didn't keep the diagram, but I will explain. Okay. There are two types of synapses which you will find or which you will divide are based upon the function. Based upon the function, that is called as electrical synapse and chemical synapse. So what is this? How you are classifying this one? Because what is the length of this gap? Yes, we have discussed about the junction. There is a gap between this. This is one neuron, and this, I hope you are able to see my fingers. This is one neuron, and this is one neuron. There is a small gap. There is a small gap. Based upon this gap, we are divided. But there are two types of synapses based upon the function. What is the main function of synapse? Transmit the action potential from one neuron to
one neuron to another neuron.
But electrical synapse means the gap, if it is below the 2 nanometers, if the gap is below the 2 nanometers, then the function or the transmit of the conduction based upon the, what is that? Connexins proteins are gap junctions. I think you know that gap junctions in the cell biology. Okay, same thing. There are some proteins which you will find in the gap junction that is called as connexin. Connexin 1, connexin 2, connexin 3. I mean to say that the action potential will pass from one neuron to one neuron in this synapses. What is that? By using the gap junctions. By using the gap junctions. This type of, what is that, junctions, you can call it as electrical synapse. Electrical synapse. Gap is 2 nanometers only. You cannot see that. What is the 2 nanometers means 10 to the power of minus 9. That's why this is called as very, very less. Okay. This is called as electrical synapse. Then chemical synapse. If the gap between these two synapses, if it is more than 20 to 40 nanometers, more than 2 nanometers, generally the chemical synapses you will find 20 to 40 nanometers. Then you will find that there is no chance of transmit this signal through the connexins or through the gap junctions, because the gap is more. That's why you need to convert your action potential or electrical current which is passing through axon into chemical signal. This chemical signal in the synapses will activate the next neuron. That's why this type of synapses you can call it as chemical synapse. I mean to say that transmission of action potential, that is the main function of the synapse. Based upon that, synapses is classified into electrical if the synaptic gap or synaptic cleft length is 2 nanometers, if it is more than 2 nanometers, more or less 20 to 40 nanometers, then it will be chemical. It will be chemical. I hope you understood the concept. Then
Yes.
coming to the next diagram. Yes, this is a synapse. This is synapse. You see that? This is a axon of the or axonal bud or synaptic bud of the one neuron. Then this is the next neuronal membrane, postsynaptic membrane and presynaptic membrane. This gap you can call it as here you can see this gap. This gap you can call it as a synaptic gap as a synapses. If the gap length is 2 nanometers, then it will be electrical. If the gap is more, then it will be chemical. Okay, and if you see the structure of this, what is that synapse? You can see that we'll discuss in the next class because already this is 3. I don't want to waste your next class time.
Sir, please one request, sir.
Yes, tell me.
సార్ మీకు పాసిబిలిటీ ఉంటే ఈవినింగ్ తీసుకోండి సార్. మాకు థర్డ్ పేపర్ అసలే కాలేదు క్లాసెస్. మీరు బాగా చెప్తున్నారు.
We will discuss later. Okay, we'll discuss later. You call me. Okay, thank you. Thank you so much. I'm stopping here.
Thank you, sir. Thank you very much, sir.
Thank you. Thank you, Raju. Thank you, all of you.
Thank
I'm
you, sir.
stopping the class. Yeah.
థ్యాంక్ యూ సార్.
Hello.